Hi guys, it's Miss Faye, and today we're going to be talking about DNA structure and function. By the end of today's lesson, these are the learning targets that we're going to be covering. And the main thing that I want you to focus on is the idea that, or basically, what is the structure of DNA and what is the function of DNA? What are the main structural components? What is the main function? We're also going to be looking at a concept known as Chargaff's base pairing rules, which you should be familiar with. Now, when we talk about DNA, we generally think of it, when we visualize DNA, we generally see it as the shape that we see here on the screen, which is known as a double helix. Okay, so this shape is described as a double helix. It almost looks like a twisted ladder. And this shape, or the first two scientists that were credited with basically showing us what DNA looks like by creating this 3D model, but also publishing a paper about the shape of DNA, are Watson and Crick, which did this in 1953. However, from the video that you watched during the Engage segment, you also learned that they would not have been able to do this without the research of another scientist known as Rosalind Franklin. And Rosalind Franklin did a lot of research with X-rays. She took a lot of images, or basically radiography images. And what she did was she had mathematical data from her studies that Watson and Crick then used in order for them to actually be able to visualize and create that 3D model. So although they were doing research on this topic for a while, her research, or that picture, or that mathematical data that was provided by Rosalind Franklin, was the missing link that allowed them to complete their research and basically come up with a 3D model of the DNA and proving that it's a double helix shape. Now, why is the DNA in a double helix? Why does it have this shape? What's the significance? Let's think about this. We know that the cell is extremely tiny, right? And if we were to basically go back to how cells look like, we know that we have the cell, right? And inside the cell, we have the nucleus. And if we were to zoom in on the nucleus, if we looked into the nucleus, then we would see that it has all of this spaghetti-like looking structures, which is basically the DNA. Now, DNA, if we take all the DNA that is found inside our cells, or inside a cell, and we lay it down on the floor, it would be around 2 meters long. So it's really, really long. It's really, really large. How do you then take something that is that big, 2 meters wide or 2 meters long, and fit it in something that is microscopic like a nucleus? Now, because it's a double helix shape, it is able, or we are able to basically take the DNA, and it's able to then twist and condense using proteins to help it become smaller and smaller and small enough in order to fit inside the nucleus. So the fact that it's a double helix allows it to twist, okay, on itself, wrap around certain proteins, condense, and become really tiny enough to fit inside the microscopic nucleus. So that is the significance of it being a double helix shape. It allows it to fit inside something as tiny as a cell. All right, now basically, if we go back to what we were discussing in the past, we know that, let me go ahead and clear this. We know that all molecules that are found inside our bodies are composed of building blocks, which are known as monomers. Now, DNA is also a macromolecule. It's actually a nucleic acid, if you remember this, right? And we know that nucleic acid is the polymer. It's the polymer. So therefore, what is the monomer of nucleic acids? We know that it is a nucleotide. So DNA, which is a nucleic acid, is made from nucleotides. That is the building block. That is the smallest part of a DNA, and it's actually made from a long chain of these nucleotides. Every single nucleotide is identical. If we look at the structure of a nucleotide, it has three main parts. It has a phosphate group, a deoxyribose sugar, so a specific kind of sugar known as deoxyribose, and this sugar is five-sided. So it has, if you look at the structure of it, it has 5 carbons, 1, 2, 3, 4, 5. And the last thing that all nucleotides have are nitrogen-containing bases. Now, you need to be familiar with the basic structure of a nucleotide. You need to know that it has 3 parts. If I was to give you a diagram, you should be able to label it. You should be able to know that it has a phosphate group, specific kind of molecule, a specific sugar known as deoxyribose, and that's where the D comes from in DNA, okay? It's because of this specific sugar. And then it has a nitrogen-containing base, okay? Now, although all nucleotides are identical in the fact that they have these 3 parts, depending on the type of nucleotide available, this will vary. What do I mean by that? Let's take a look here. So there are 4 variations of these nitrogen-containing bases. Thymine, cytosine, adenine, guanine. What do I mean by that? I mean that this nitrogen-containing base could be for one of 4 things. It could be a thymine, it could be a cytosine, an adenine, and guanine. And this is why we have variation. Even though our DNA, all organisms on Earth have the exact same makeup of their DNA. All DNA is made from nucleotides. The reason why we have variation is because these nucleotides come in these different varieties. So depending on which nitrogen base they have, you'll either end up with a thymine, a cytosine, an adenine, or a guanine. And the order that you have these bases in, so the order that these bases are present in your DNA, will also contribute to the variety that we see. So what do I mean by that? Basically, if we were to take the DNA double helix, if I was to take it and I was to lay it flat, it would look something like this. So remember, it looks like a ladder. And these steps that you see, this used to be twisted, by the way. So I basically took this thing, all right? Sorry, my pictures are kind of wonky, but I'm doing my best. Okay, so if I take this twisted ladder and I laid it flat, this is what I would have. And essentially all of these steps that you see are the bases. So for example, I might have A, C, T. A is adenine, C is cytosine, T is thymine. Another person might have CTA. So even though we both have the exact same nucleotides, the variation of the nucleotide or the order is really going to contribute to this genetic variation that we see all around us. Now, the nucleotide themselves, I don't want you to worry too much about what's going on this slide, but I want you to see that thymine and cytosine are very similar in the fact that they have a structural component of being single ringed, meaning that they are made from, or their structure is made from one ring, okay? So they have the one ring structure. On the other hand, adenine and guanine have 2 rings in their structure, okay? So they are similar in that sense. Why does this matter? Why does this matter? Let's take a look at another scientist by the name of Erwin Chargaff in 1950. He did a lot of research and he kind of analyzed DNA from many different organisms. And then he started to notice a pattern. He started to notice that all the organisms, all their DNA, no matter what the organism was, they all had these bases showing up in their DNA, and they were the same 4 bases. But the proportion, meaning how many of each base you have, differed. And then he noticed a relationship. He noticed that the amount of adenine always equaled the amount of thymine, and the amount of cytosine always equaled the amount of guanine. No matter the organism, no matter what DNA he was looking at, he always noticed this kind of trend, okay? So he noticed that the amount of adenine always equaled the amount of thymine, and the amount of guanine always equaled the amount of cytosine. This was interesting. So he kind of took this information, and then what he did is he came up with a rule. Okay, he called it Chargaff's rule, after his name. And this rule says that, this is known as the base pairing rule, and it says that adenine will always pair with thymine, and that's why you have equal amounts of them. And then cytosine always pairs with guanine, and that's why you have equal amounts of them. What do I mean by that? Let's go back to the picture of the DNA that I drew earlier, the flattened DNA. So let's say that I have a DNA strand, and of course it's a double helix, meaning that it has two sides, okay? And we have the bases right here. These are the nitrogen bases. These are the nucleotides, the building blocks, and then this is the whole DNA molecule. Let's assume that I have the following bases: adenine, cytosine, guanine, thymine. According to Chargaff's base pairing rules, adenine will always pair with thymine. Cytosine will always pair with guanine. Guanine will always pair with cytosine. And thymine will always pair with adenine. Okay? This way, you will always have the same amount of adenine and thymine and cytosine and guanine in any strand of DNA that you're looking at. Let's look at a mathematical problem. Okay, I promise it's nothing too crazy. If I can solve this, then you're good. All right, so basically, let's look at the first question. And what I want you to do is, before I go over the answer, I want you to pause and try to attempt the question yourself, okay? So the first question says, a DNA molecule has 2,500 guanine bases and 3,500 thymine bases. How many DNA molecules does it contain? So let's kind of break it down. We know that DNA is a nucleic acid, and it's made from nucleotides. And we know that nucleotides come in four varieties: adenine, cytosine, thymine, guanine. Now, the question is saying that we have 2,500 guanine bases, so I'm going to go ahead and put 2,500 here, and 3,500 thymine. Now, according to the base pairing rules that we already learned about, we know that thymine and adenine are always going to be equal. So therefore, how many adenines do we have? We can deduce that we will have 3,500 adenines, because adenine and thymine are always going to be equal. And then how many cytosines are we going to have? It's going to be equal to the amount of guanine bases, so 2,500. If we were to add all of these bases together, then we would end up with 12,000 bases, or BPs, base pairs of DNA. That's how much DNA we have, okay, in this particular molecule. So that's the answer for the first question. Let's take a look at the second question. The second question says that a fragment of DNA was found to have 120 guanine bases and 60 adenine bases. What is the number of cytosine and thymine bases? Easy, right? So again, we know that DNA has 4 bases: cytosine, guanine, adenine, thymine. And it's saying that we have 120 guanine and 60 adenine. Now, according to the base pairing rule, we know that adenine and thymine always pair. So therefore, we must have also 60 thymine. And we know that cytosine and guanine always pair. So we must also have 120 cytosine. There you have it. Okay. All right, now when we're looking at, when we're looking at DNA, we said has to condense at some point, right? That's how it's able to fit in this tiny nucleus. And it does that by wrapping around specific proteins. And these proteins are actually known, they have a specific name, of course, because everything in biology has to have a fancy name. These proteins are known as histones, okay? Now, at the beginning, so if you remember, if we go back to the cell cycle, right, we said there's several phases or stages. The first is interphase, so that's like the main part of the cell cycle, and that includes G1, S, G2, right? This is when the cell is kind of preparing to divide. And then the second part is M, which includes mitosis and cytokinesis. Now, during interphase, DNA is not dividing, right? It's just kind of preparing to divide. So DNA is going to be found in this very loose, almost like spaghetti noodle looking form, okay? This loose form of DNA is known as chromatin, all right? So when the DNA is in this loose, uncondensed form, it's kind of like preparing to divide, it hasn't really divided during interphase. It's known as chromatin. And chromatin it can be associated with these specific proteins, right, called histones. So you can see histones right here. They look like these purple grapes. And essentially chromatin, which you see right here, this is chromatin, the blue strand, is gonna wind and wrap around it, and then you'll end up with something that looks like that. Okay, so this is chromatin, is basically the DNA that has been wrapped around the protein histone, and it's kind of like still in its loose form. Now, when the cell is ready to divide, it's going to start to thicken, okay? So essentially what's gonna happen is that it's going to turn from looking like this, very thin, it's gonna start to thicken. And when it's in its thickened form, we refer to it as a chromatid, okay? So the thin form is a chromatin. The thickened form is a chromatid. And then during cell division, or before the cell even divides, we know that in S phase, what happens? What happens to the DNA in S phase? It gets duplicated. So essentially, this strand, this chromatid, ends up like an identical copy ends up being created. So you'll end up with two chromatids. And they're usually shown as looking like an X, right? Something like this. This is known as a chromosome. Now, two of these together, remember, this is a chromatid and this is a chromatid. Together, they are known as sister chromatids, okay? And remember, they are basically identical copies of each other, because what happened is in S phase, whoa, what happened there? Okay, that was supposed to be a circle. In S phase, this chromatid ended up being duplicated, and an identical copy was created, and you end up with this chromosome. So make sure that you know how these things are related. Make sure that you know how chromatin, chromatid, and chromosomes are related. So basically, to summarize, a chromosome, like we said, is a thread of DNA, and it has these areas on it that are called genes. It's tightly packaged, it's condensed, it's thickened, just like we saw in the previous diagram, and it's only visible, this is important, it is only visible during cell division. So basically during mitosis and cytokinesis, we can see this thickened form of DNA. So if you were to observe a living cell through different stages of the cell cycle, you will be able to see DNA much clearly under a microscope, of course, when it is during cell division than during interphase. Like in interphase, the DNA is kind of like not visible because it's too thin. Remember, it's in the chromatin form. But then during cell division, it's going to thicken, it's going to condense, and therefore you should be able to see it much easier underneath the microscope. Now, if we take a chromosome, all chromosomes contain these areas, okay, segments, sections. A section of a chromosome is known as a gene. Okay, is known as a gene. Each gene contains instructions for making a specific protein. Remember, proteins make our body live. They basically are responsible for pretty much every part of your existence, from your hair shape, to your eye color, to your, skin color, to literally every characteristic, every trait that we see physically, and even things that are not physical, things like transport, all these mechanisms that happen inside the cell, they are caused by the help of proteins, okay, or they happen because of the help of proteins. So essentially, the chromosome, which is basically DNA, condensed DNA, contains sections on it, as you can see in this picture. And these segments, these sections are known as DNA. Genes, okay, these sections are known as genes. And each gene has the instructions for making one protein, okay, each gene, and you have hundreds. You don't just have one gene on a chromosome, you have hundreds of these genes basically lined up all on the chromosome. So they're responsible for basically carrying the instructions for making proteins. In addition to making proteins, we already talked about that. DNA also has other functions, which we'll talk about next. But before that, I just want to, again, review what chromatid is. We already talked about that. We already mentioned what sister chromatids are, and we said that sister chromatids are basically two sides of a chromosome, right? So this is a chromatid, thickened form of DNA. This is another chromatid. Together, these are sister chromatids. And again, just like it says here, they are identical because they're basically copies. A chromosome is held at the center by this structure or this area known as the centromere, which makes sense, it's in the center. And the last thing I want to talk about in terms of the structure of DNA is the telomeres. Now, telomeres are super interesting, and basically they are found at the edge. They are like these caps that are found at the ends of the chromosome. Okay? If you have shoelaces, you'll notice that at the end of a shoelace, you have this like plastic cap thing at the end, okay? That's my shoelace. And what's the purpose of that plastic cap? Basically, it's to prevent the shoelace from fraying, from being damaged, from coming apart, right? Now, the telomeres act very similarly. They basically protect the DNA from fraying, from being damaged, from shortening. And unfortunately, every time the cell goes through a cell cycle, division, so every time the cell divides, and remember, cells divide so many times, depending on the type of cell. Some cells divide pretty much your entire life. Some cells divide for weeks at a time. It really depends on the type of cell, but every time a cell goes through a cell division, the telomere is gonna get a bit shorter, okay? It's gonna get a bit shorter, and then a bit shorter, and a bit shorter, and then eventually it basically almost becomes non-existent, and the DNA is gonna start to be more prone to being damaged. And this is when we start seeing symptoms of aging, things like diseases, things like organ failure, wrinkles, gray hair. All of these symptoms of getting older are basically due to the fact that the DNA is slowly getting damaged because the telomeres are getting shorter and shorter and shorter, and there's nothing really there at the ends to protect the DNA during cell division. So theoretically speaking, if scientists can figure out a way to prevent telomeres from shortening and keep them protecting the DNA, we could potentially stay young forever. And that is obviously a very tempting and interesting topic that scientists and researchers are constantly trying to understand, in hopes of extending the human lifespan, but also extending, not only living longer, but also having a better quality of life when you're living longer. So staying younger longer. There's a lot of interesting things that you can read more about when it comes to telomeres. I highly recommend you research more about it. It's a very interesting structure and topic. All right, last but not least, we're going to talk about the function. Very straightforward. We talked about so much of this. But basically, if we were to break down the function of DNA into 3 parts, it obviously carries genetic information. It carries hereditary material, meaning that it carries information that can be passed from parent to child. And finally, like we mentioned, it carries instructions for making proteins. Every gene, which is found in a chromosome, which basically is DNA, every gene is responsible for making one protein. I hope that was helpful. As usual, if you have any questions, please feel free to ask. Have a great day.